am here to talk to you about my perspective on global amphibian declines. This is a phenomenon that has been happening for, uh, we first learned about the mysterious, enigmatic declines in about 1999 when I was a school kid in Zimbabwe playing with wildlife. I I lived in Zimbabwe and really just love nature and like to get outdoors. But a lot of people are very surprised to learn when I tell them that I would cycle literally past elephants and zebras on my way to school, and I would dream about salamanders. I would dream about the biodiversity hotspot of salamanders, that is Appalachia, because that was so exotic to me. And there were no salamanders in Zimbabwe. And I had some pet axolotls, and I thought they were just very cool creatures. Um, but but my career did lead me into trying to figure out ways in which we can mitigate threats to these lovely, incredible creatures all around us, right? And so that's how I define conservation: is how do we use the limited set of tools in our conservation tool chest um, to mitigate threats. So when I did my PhD in the British Virgin Islands, I studied um, biodiversity patterns on coral reefs. And what I'm doing here is I built over 100 different artificial reefs to see which kind of configurations would have the most biodiversity, right? Is it the number of growth forms? Is it the variety of hull sizes? Is it the hard substrate? Is it the height of the reef above the substrate? Which variables in a multivariate problem are really driving fish biodiversity at a very local scale? But that was a very ecological problem, right? I wasn't really studying how to stop the main threat to coral reefs at the time was climate change. It still is. At the time when I was working in the British Virgin Islands, we had about 10% live coral cover. I went back right before COVID and had a look, and it was about 0.5% live coral cover left. So that decline happened in the 20 years since I finished my PhD. So those threats are still happening. But then I went to work for Save the Tiger Fund at the National Fish and Wildlife Foundation. And this job was literally giving out money to people working in 13 tiger range countries. And my job was to figure out, how do we take this limited pot of money and get our biggest bang for it? How do we save the most tigers with it? And so in doing that, I learned a lot about conservation. I learned that you can conserve things by setting aside land for nature. You can try and change people's behavior. Instead of using tiger bone medicines, they could use alternates to tiger bone, right? There's a lot of different ways. There's a lot of different tools in our conservation tool chest. So that really kind of illustrated for me what came to define the term as conservation, and myself as a conservation biologist. So as a conservation biologist, I study the limited universe of threats that act on all species and ecosystems. And I also study the limited set of tools that we have in our tool chest to be able to mitigate those threats. So one of the things you quickly learn in this field is, the most basic tool we have in conservation is setting aside land for conservation. Yet you go to any conservation course in the United States where they're charging you a lot of money to teach you how to be a conservationist, and you ask any of those professors, how do you get 1 million acres set aside for conservation? And they'll just shrug their shoulders. And that's our most basic conservation tool. Yet we don't really, a lot of people don't really know how to do it. So that is what I've been working on. Now we'll just see if we can get to the next one. So recently, you may have seen in 2003, we released the most recent assessment of amphibians. And what we did is we got about 1,600 herpetologists from all over the world. And we did it country by country by country and species by species by species. And we assessed 8,000 species in the conservation status. And what we learned is that about 40% of all the amphibian species on the planet are threatened with extinction. We had a fair idea that that number was in that ballpark from the previous assessment about 20 years ago. But what was happening about 20 years ago is a little different to what's happening now. And the things that seem to be taking over as threats right now seem to be really in the climate change area. That was not so much on our radar screen about 20 years ago, but now it's becoming increasingly a concern. So what are we going to do about it, right? So that same group of people through the IUCN Amphibian Specialist Group got together and said, okay, we're going to develop a roadmap for amphibian conservation. So we developed this IUCN Amphibian Conservation Action Plan that will be coming out. Should have come out at the same time as this paper, and it's still not out yet. So it should be out shortly. But it's divided into various different chapters with folks saying, okay, this is what we can do about it. But what have I been working on? Sorry, I'm going to go back a bit. What have I been working on? When I started my job at the National Zoo, I came from the National Wildlife Foundation, and I was given a desk and a computer and 6 months of salary and told to go save the frogs. And I was like, okay, I don't have any budget. And frogs are in deep trouble. What are we going to do? And at the time, there was this disease that was spreading through Panama that had been sweeping through Latin America and was associated with a lot of these enigmatic declines that were first discovered in the 1990s. And so this amphibian chytrid fungus had been sweeping through. And in Panama, one site lost about 90% of the frogs and half of the species in 6 months after this arrived. Yet at the Smithsonian, we have the Smithsonian Tropical Research Institute, which has about 6 research facilities all along the Panama Canal, because that was former US territory. And we have this legacy of this infrastructure that now serves about 400 visiting scientists every year. And I said, well, there's something there in Panama that the Smithsonian has, that there's some resources we can leverage to help solve this problem. So if this is the big threat and this is where it's happening, where Smithsonian has some capacity, this is where we need to be working. And it also is kind of, the story is kind of closed in the loop, because the amphibian chytrid fungus was discovered in 1999 at the National Zoo from the Reptile Discovery Center, when we had some Dendrobates tinctorius die, and our pathologists were looking at it and they said, oh, well, there's a strange thing growing on the skin and we're not really sure what it is. Is it a protist? Is it a fungus? Is it a bacteria? What is it? And they were scratching their heads over this. And then they called up an expert on chytrids, and the world's only expert on free-living soil fungi was a lady by the name of Joyce Longcourt at the University of Maine. And she's like, yep, this is a chytrid. Where did you find it? And they said it's in frog skin and it's killing frogs. And it's like, that's super interesting, because chytrids aren't parasites. And so she was super piqued by this and helped to describe this new organism, Batrachochytrium dendrobatidis. And what that literally means is batracho frog. Chytrium means flask shaped. So you see the sporangia here is sort of like a pot. And dendrobatidis, because we found it in a Dendrobates dart frog. And so when that amphibian chytrid fungus was described, people started gradually piecing together a lot of these mysterious amphibian declines from around the world were actually connected with this pathogen. And since then, we have determined that about 90 species have disappeared altogether from the chytrid fungus, and about 124 species have declined by more than 90%. So just a little timeline here. In 1999, we had the amphibian chytrid fungus was described. And at the same time, a researcher in Panama, Karen Lips, was noted that these frogs were disappearing. So we imported golden frogs, Panamanian golden frogs, from Panama to the United States to keep in captive assurance colonies to ensure that if this disease did come, that we'd still have some breeding. In 2004, the chytrid reached central Panama, which is the home of the Panamanian golden frogs, causing mass mortality. The Houston Zoo began constructing a facility in Panama to help rescue some of these frogs. And in 2009, when I was hired, we came into the picture and joined with the Smithsonian and other zoos to form a countrywide Panama amphibian rescue and conservation project. And so this partnership between various zoos and the Smithsonian is essentially to establish captive sustainable assurance colonies of amphibians in danger of extinction from the chytrid fungus, and to figure out a cure for the disease and figure out how we can reintroduce these frogs back into the wild. So that's the mission of what we're working on. So the first goal was, okay, we need to actually build an amphibian ark, right? And we need to train the people how to keep the amphibians. And so we had a team of folks at zoos and aquariums and at the National Zoo who were really good at looking after and breeding these amphibians. And so they came down and they helped us to build this facility. And when you look at the facility, what we have these shipping containers that were going up and down the Panama Canal, and I didn't have any money, and I saw all these shipping containers. I said, you know, if they can keep strawberries frozen up and down that Panama Canal, they can keep frogs at the right temperature inside a tropical climate. So I called up Maersk Line and I said, Maersk, you got to give me some of these pods so I can use them for amphibian rescue pods. And they were like, no, go away. So eventually, as happens in DC, I used my network, and eventually one of my lawyer friends said I should talk to their lobbyists, the shipping lobbyists in town. So I called up the World Shipping Association, and they didn't even bat an eyelid. They just said, how many pods do you want and where do you want them delivered? We'll have them there next week. And they thought this was just a no-brainer. And we got these Maersk to donate 7 of these shipping containers. And we built an amphibian ark. So we have 7 of these shipping containers to house frogs. We have some office space, a lab, a quarantine facility. We have a place for breeding food to feed all of these animals. So this is actually one of the largest dedicated ex situ conservation facilities for amphibians in the world. And we employ about 11 full-time husbandry professionals. We've trained about 10 Panamanian and 8 international students or fellows and more than 70 interns in the last 5 years. So we've been having an impact on the human capacity on dealing with these problems. So that is what we've been working on. But this is the faces of all of the creatures we've been trying to save. We prioritize these out of 200 species of frogs in Panama. That's about the same number of species as are in the United States altogether, right? In one tiny little country in Latin America. And we said, which are the species where we have a chance of breeding them successfully at captivity, where they're likely to go extinct in the wild, and we can avoid a species extinction. And these were the ones we prioritized. And so now we manage about 2,000 adult animals from 12 different species. And so when you bring animals into a captive assurance colony, we really got to bring in enough animals to ensure the genetic integrity of the species over 100 generations in captivity. So the minimum numbers you're really looking for are about 12 male and 12 female pairs represented in the next generation, which in practicality, because you often don't breed every single individual that comes in as a founder, in practicality, that means you need about 20 males and 20 females for each species. And so a lot of these animals were very rare anyway, or had declined. And so we had to go out into the forest and rescue these animals. We've also been working on, sometimes these frogs won't breed when you get them into captivity. So one of my colleagues here, Gina Della Togna, has been developing hormone treatments so we can get these animals to lay eggs or produce sperm with a hormone treatment. And she's developed protocols for collecting and cryopreserving sperm from about 7 species. And so we've also been banking frog sperm in the hope that, if we do lose some of the genetic integrity of these species in our captive lines, that we'll still be able to have some frozen germplasm that we can access to rebuild that. I work at the National Zoo out of Front Royal, and we've done that exact same thing with black-footed ferrets in the United States. We've had one of our black-footed ferrets reproduce 20 years after he was deceased, and contributed his valuable genes back into the living population because we had banked some sperm. So that's just one of the ideas. The other thing that I've spent most of my academic attention on is trying to find a cure for the amphibian chytrid fungus. So we've been looking at various different things to try and mitigate this threat, right? Because putting animals in glass cages and keeping the fungus away from them is not mitigating the threat. It's buying us time. And so we need to be doing both, right? And so we've been, the first thing that I did was, this experiment was actually quite interesting that you see here. We took the most common skin bacterium growing on all golden frogs, and then we worked with a team out at MIT, and we genetically engineered those bacterium to produce antifungal chemicals. So the idea was that these frogs, when they had this bacteria growing on them, would be growing their own anti-chytrid pharmacy and protect them from the chytrid fungus. We got the bacterium engineered beautifully. We put it back on the frogs, and it didn't persist very well on the frogs, and we did not have any protective benefit, but we tried really hard. We've also tried, more recently, we've tried a vaccination trial. Some folks have seen in some species that if you expose them to the disease, clear them with the antifungal chemicals, and then re-expose them, they have some acquired immunity, right? So a lot of vaccinations will give you an attenuated vaccine, an attenuated strain of whatever pathogen that your body can easily fight, but it's the same immune response for the attenuated pathogen as the full version pathogen. But what we found for particularly the harlequin frogs, the beautiful little toads, that there's about 100 species of them in the neotropics. They're super susceptible to the disease, and we were unable to develop a vaccine. The ones that had been pre-exposed to the disease actually died quicker upon re-exposure. So we are now working on just trying to understand what are the immune mechanisms around their reaction to this pathogen, and how does it compare with the immune mechanisms of a species that can produce a successful immune response. So we're really trying to understand in very fine detail what is the pathology of this disease, and how does it interact with these organisms. So with the goal that we want to breed resistant frogs. So one of the things we've been doing more recently is if you take a sample of the skin mucus from these frogs and then put the amphibian chytrid fungus just in the water that contains that mucus, you can look at how the the zoospores live or die in that solution. And that can give you an indication of how anti-chytrid that skin mucus is. So the idea is that maybe we could breed frogs, breed the individual frogs with the most powerful skin mucus to make a more resistant frog, just like you'd breed a cow to produce more milk. So one of the other things we've been doing is, because a lot of these frogs have disappeared, we've been trying to figure out how do we monitor for frogs that just aren't there. And it's quite tricky. One of the frogs that disappeared, sorry, I've got to shut my dogs down. It's okay. One of the frogs that disappeared was called the Rab's tree frog. And it's a frog that calls about 9 times a night. And it calls from about 30 meters up in the rainforest canopy. So they're not easy to find. And we said, well, how do we find this frog? How do we even know if it is extinct? So we deployed these automated recorders out in the rainforest. They record 1 minute every 10 minutes. And we used artificial intelligence to do pattern matching to listen to about 70,000 recordings. That's like 50 days worth of continuous listening if you were an intern stuck on this problem. Fortunately, we got AI to do it rather than an intern. And we were able to do pattern matching for calls of a bunch of different frogs, common species and some of these lost frogs. We didn't find the Rab's tree frog, but we did find this one, which is Tryprion spinosus. It's labeled Anatheca spinosa because that's just the program's using the old name. But what we did is we recorded this call and did a sonogram of its call from inside our shipping containers on the right. And then we recorded in the rainforest, and we noticed that at the same frequency, at the same sort of interval, there was this call. And when we listened to it and we looked at the time stamp, we figured this must be Anatheca spinosa, which was last seen in the wild in Panama in 2015. So that's kind of cool, and it means we can probably do some habitat supplementation at that site and maybe release some of our captive animals into those to do a population supplementation there. We've also been, this was one of our first reintroductions of golden frogs. We tied these little radio transmitter backpacks to them so we could watch what happens when you release a frog from captivity back into the wild, and just understand where did they go, what did they do. And what we found is that the females dispersed a lot further than the males. We also found that if you house these animals inside mesocosms for 2 weeks before you release them, they didn't disperse as far. And if they don't disperse as far, they have lower mortality, because if you're not running around like crazy, there's less chance you're going to run into a scorpion or a spider or a snake that's going to eat you. And your predation rates are a little lower. So this was not a, we have not seen any persistence of these animals more than about 2 months after we've released them. But we've now done 3 release trials with these animals. We've learned a lot about how these animals transition from the wild, from captivity back into the wild. And so now part of our research is going to be to look into whether these frogs, this is a mesocosm here on the right, and this is one of my PhD students tying a radio transmitter onto these golden frogs. So what we want to do is figure out, when we release these frogs back into the wild, they get eaten. There's just a huge amount of predation. So they're getting chumped up before the chytrid can even get to them. So we haven't solved the chytrid problem, but we've also got to solve this predation thing. And one of the reasons that they get eaten is because these are poison dart frogs, and they are aposematically colored. They have this warning coloration, so they attract predators. And then if the predator eats them, they're supposed to be toxic, and then the predator will spit them out and remember never to try that again. Now, what happens is in captivity, we've been raising them on a diet of fruit flies and crickets that do not have any of these skin toxins. So there's no toxins for them to sequester. So we're releasing them without their skin defenses. So we need to figure out how do we restore their skin defenses. So we've actually, we have a project where we've tried feeding these animals a little worm that's been injected with enough tetrodotoxin. This is the same kind of toxin that's in fugu pufferfish that you even get a little bit in your mouth. You can die from it. We've injected one of these worms with enough toxin to kill 1,000 mice, and then fed it to the frog. The frog was just quite happy and looking for more. No problem whatsoever. So now we want to see how much of that toxin it's sequestered into its skin. But this is very, very advanced research, and doing work on a chemical that toxic involves a lot of paperwork, especially getting that chemical into Panama and having people appropriately trained to handle it and use it. So this is not easy stuff, but we will continue the work and the challenge, because we have to. We are in a very important position of a huge amount of responsibility, and it weighs on us daily, right? We have the future of 12 species in our hands, no one else's hands. No one else can do anything about it, and we've got to figure it out. So we're going to do our darndest. And part of that is trying to persuade people that frogs matter, right? And so if any of you have been to the National Zoo in Washington, D.C. Amazonia exhibit, you may find this image of this hexagonal tank very familiar, because it was modeled off of our Amazonia exhibit. We took this exhibit to Panama, and we have it using Panamanian species in Spanish and English. But you can see here that what is important is not the signage or the graphics or the blah, blah, blah. Those kids are seeing that these frogs are cared for. And by seeing that we're paying so much attention to them, that we're caring for them, they understand that they're valuable, that they're important. And you're not explaining that to them, you're showing it to them. And I think this has just been a super powerful way to reach the public in Panama. And we also have a little exhibition at our frog center. So if you're in the town of Gamboa and you want to take a peek inside an amphibian rescue pod, you can go and look inside a working amphibian rescue pod. We are trying to do some fundraising right now so that we could actually open that pod to the public so people could walk through it. But that is just going to have to be deferred to next year, maybe. But it would be very nice to be able to actually take the public into the pods, because right now, using our biosecurity measures, we've got to be very careful about who goes in and how they go in and how their shoes go off and all of that kind of stuff. So we want to keep the amphibian chytrid fungus out and keep the frogs safe inside. And every year on August 14th, we have about 2,000 school kids in El Valle de Anton, which is the home of the Panamanian golden frog, dress up like Panamanian golden frogs and walk down the streets of Panama and say, save our national heritage, save the La Rana Dorada. And we've got a lot of awesome outreach materials. We had a Colombian musician write a song, La Rana Dorada. It's very catchy, it's very cool, and did a little music video. And so we've had a lot of real honest to goodness community engagement, and people do love their frogs in Panama. So at this time, I normally kind of just show this and I say, well, how do you communicate values to people, right? And how do you value the toad mountain harlequin frog, which is a frog that lives in a very tiny little mountain right on the border of Panama and Colombia, and is probably extinct in the wild now. And we have every single individual of this species on the planet sitting inside one of those shipping containers. And how do you say which is more valuable? The Jackson Pollock #5, which sold for $140 million in 2006, or the Jackson Pollock frog, which looks like it's been splattered with yellow paint, that is on the verge of extinction. So this is just something that we need to think about, because the fact of the matter is the Prairie Mountain Harlequin frog probably doesn't have a lot of ecosystem service values. Its distribution is so tiny. If it goes extinct, the howler monkeys will still howl, and the sun will come up, and the trees will still grow, and life will go on. And but why what makes a Jackson Pollock painting valuable? It's very unique. It can't be replicated. It is something that cannot be forged, right? So there are a lot of kind of similarities, and these are the tangential things. We don't have to find an ecosystem service value for everything. We don't have to find um, monetary value for everything in order for something to have monetary value. And I think that this Prairie Mountain Harlequin frog, if you think about it carefully, does have some of the attributes of a work of art. So with that, I want to change gears a little bit and get into some of the local amphibians. And seeing as this is the Maryland group, Maryland is a salamander haven with 22 salamander species and 20 frog species. In Virginia, we have 50 salamander species and about 20 frog species. But the Appalachian region, as I told you in the beginning, is a biodiversity hotspot for salamanders. So if you were to look at this map of the world and try and say, okay, if you wanted to figure out what makes North America special as a continent compared to any other continent, it's our salamander diversity. It's not that we have bison. There's bison in Europe. It's not that we have bald eagles. We get fish eagles in Zimbabwe. I used to see them all the time. Not a problem. But it's the salamander diversity. And this is a really unique thing. And in the United States, amphibians aren't declining as fast as we've seen these chytrid-related declines in the tropics, but some species have disappeared in relation to chytrid fungus. I believe the green salamander in the eastern United States has disappeared because of chytrid fungus. The Wyoming toad, the mountain yellow-legged frog, the Oregon spotted frog, these have all declined because of the amphibian chytrid fungus. And there are other reasons why amphibians are declining, not just this fungus. And just at a general rate, we're losing about 4% of our amphibians per year. And that's from a USGS study. So I work out at our campus at Front Royal. We participate in a project called SparkNet, where we monitor redback salamanders. And so we have these plots with 50 cover boards, and the students will come and help us collect the salamanders. And then each of these salamanders, we will give a unique mark to. So you see these little fluorescent tattoos, and each one gets a unique color code. And that way we can figure out who's who. And then through a marker capture method, we can determine how many salamanders are out there that we didn't see. So it's a simple ratio thing. And so what we found with our boards at Front Royal is there's about 2.2 redback salamanders per square meter of forest out there. And that sounds okay. That's fine. 2 little redback salamanders in a square meter. Yeah. But when you add that up to our 600 hectares of forest, that's 12 to 16 million redback salamanders just on our property in Front Royal. So they are very, very abundant vertebrate, and they're very cool. And so it is important to do these kinds of monitoring things, because not all of the amphibians out there or salamanders out there are doing so well. So hellbender salamanders are our largest salamander in the United States. They're one of 3 species of giant salamanders in the family Cryptobranchidae. And that includes Japanese giant salamanders, Chinese giant salamanders, and then hellbenders. And so these hellbender salamanders have disappeared from about 300 of their 600 known sites. So we've lost about half of all of the known hellbender sites. And then at the actual sites where they are persisting, many of them are declining. And one of the things that people have noticed is they're failing to recruit juveniles. So some of these places you'll only find big, old salamanders and no babies. And some places you do find babies, but some of the places you're only finding these old salamanders, and they live for a long time. So the question is like, why are they not recruiting juveniles? And so people have been talking about, oh, well, there's lots of threats, and the watershed, and the pollution, and the siltation, and the blah 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 blah. So there's lots of potential things that are going on. But one of my colleagues, Kim Terrell, was working on. Oh, I gotta switch gears. One of the questions out there was, have one of the reasons that these amphibians declined was because of the amphibian chytrid fungus. So when you take these salamanders and expose them to the amphibian chytrid fungus, which we did in trying to develop a vaccine for them, you can see that when the negative chytrid fungus animal looks like this, and the one that has the chytrid fungus looks bluish, and its skin is all gets thickened, and it has all of these chytrid zoospores growing in it, whereas a healthy salamander has very smooth skin that's not been thickened or keratinized. So there's definite effects of this chytrid fungus. And when we've been reintroducing hellbenders in places in New York, some of these animals have been dying, and when we examine them pathologically, they have the chytrid fungus. So the question is, are they dying from this chytrid fungus when we're reintroducing them into the wild? Do they get the chytrid fungus in the wild? And can we develop a vaccine for them? Because they seem to be fairly tolerant of the chytrid fungus under normal circumstances. So we tried actually giving them a vaccine. We tried giving them killed chytrid fungus and exposing it to them either orally or on their skin. And so what we found is that when we this is the on this graph, this is the experimental day post-vaccination, and then we expose them to the amphibian chytrid fungus. And on the vertical axis is the chytrid load that came up. And I'm not sure if you can see here, but the ones that we gave the oral or the topical vaccine to all got infected at the same rate as the ones that we gave a sham non-vaccination to. So what was happening here is that vaccinated and unvaccinated hellbenders all got infected. And they retained their infection for 220 days. Let me tell you, that is a super long experiment from a chytrid experiment point of view. And they did not clear their infections, but they managed to tolerate them. And so what we did learn was that the animals that were vaccinated did have an immune response to the chytrid fungus from the vaccination. that was similar to animals seeing the disease for the very first time. But that immune response did not protect them or give them advantageous disease outcomes. So this animal just seems to be able to tolerate the disease without dying from it. So that is an interesting finding, but vaccination is not going to help us control it. Uh, um, so we think that the chytrid fungus is probably not the reason for these declines in the hellbender populations. So this is when I'm going to tell you about Kim Terrell's work. Now she worked to see what is the effect of climate change on these amphibians. So we tried replicating the temperatures of a stream in Buffalo, New York, and in these tanks we replicated that temperature change throughout all of the seasons. And then we also did another set of tanks right nearby where we elevated that temperature. We still varied it, but we made it 1.5 degrees warmer. And what we found is that in summer, the animals that were 1.5 degrees warmer were not eating for 3 months in the summer. And so they were losing weight for 3 months in summer, whereas the animals under the current climate trajectory, they were not eating for just 1 month out of the summer, but they were gaining weight for 2 of the 3 summer months. And now, why is this important for hellbenders? Well, hellbenders, the males form dens and they guard their nests. They're called den masters. And then multiple females will come in and deposit their eggs in these dens. And then the male will guard them and will not eat and will just defend these eggs from other hellbenders that want to eat them, or crayfish or fish, and they will look after them. But if you haven't eaten for 3 months, you're probably going to end up looking at this big pile of juicy eggs and just eat them and then get on with your life. And that's what seems to be happening. And I think that climate change is leading to a failure in recruitment of hellbenders. So there's a bit of gloom and doom for you, but I have been very excited because we've been searching for all of these solutions um, for amphibians. But one of the things that you can do is you can create vernal pools, and they don't have to be big affairs. All I did here was in a wet, squishy area in my backyard. I dig a hole about 10 feet by 7 feet wide, and it it held water. And what you see here is all of these are spotted salamander egg masses, these pale blobs, greenish blobs in the water. Those are all salamander egg masses. And they are real amphibian factories. And every year I produce thousands of wood frogs, spotted salamanders, tree frogs, spring peepers, all coming out of these vernal pools. And what's so important about vernal pools is they're places without fish. And so these are all some of our local amphibians that you can find that will be breeding in vernal pools. Now, that red-spotted newt will breed in the vernal pools just fine, but he's also a predator of the other eggs of these other frogs that like to hang out in these vernal pools. But they are incredible, and we produce millions and millions and millions of baby frogs just through having some fishless habitat for these amphibians to breed in, and it's been very rewarding. I now have 10 vernal pools on my property, and I'm still going to be keep digging some more, because that is the limiting factor for these amphibians. One of these amphibians here, the spotted salamander, can live for 30 years. So it is possible that these spotted salamanders that found this vernal pool have been on my property for many years, since previous vernal pools have been drained. Because anytime anyone sees some standing water, they want to dig a ditch and get rid of it because they don't want mosquitoes. And so it's possible that some of these spotted salamanders have been sitting for a decade waiting for waiting for some vernal habitat to come back. And by digging these vernal pools, they've managed to come out and find it. And it's just been super rewarding to watch. So with that, I want to wrap up and say thank you very much for listening to me, and thank you to many of the sponsors who have been helping us with our project. It's been a real village of people who have helped us out there.
Thank you so much, Brian. Let me put a spotlight on you, and then we can open it up for questions and answers. There are a couple in the chat box from Steve. One is, and I had this also, I was going to ask, why, is there a reason why the Southern Appalachians are such a biodiversity hotspot for salamanders? Do we know why?
Well, so what seems to be happening is these post-glacial thaws, there's a lot of mountaintop endemics. So what's been happening is as the glaciers have retreated, there've been like some groups of salamanders. You may have heard of slimy salamanders or redback salamanders. Both of those have a whole complex of different, very similar looking species that only occur on one mountaintop. So, for example, I live next to the Shenandoah National Park, and on the north-facing talus slopes of Stony Man, Hawksbill, and Pinnacles Mountains, you can find the most restricted range vertebrate in the United States, which is the Shenandoah salamander. And it only occurs in like 6 square kilometers anywhere on the face of this planet. And it clearly evolved from the redback salamander. They're quite difficult to tell apart from redback salamanders. But they are genetically distinct. They don't interbreed with them. They're a little bit larger than them, and they possibly evolved through isolation on one of these post-glacial retreat mountaintops from redback salamanders. And so what's happened is there is this evolutionary engine with these mountaintops and post-glacial retreat. Now, the salamanders themselves, the most diversity, there's deep evolutionary diversity, as in cryptobranchids. So we do see like giant salamanders and other things like amphiumas and sirens and all kinds of deep evolutionary lineages of salamanders here. But we also get highly diverse plethodontid salamanders, which are the lungless salamanders, and they make up a lot of the species that you'll see in Appalachia. And so there's just been a lot of opportunity for those to evolve. Now, those ones really like these cool, moist conditions because they're lungless salamanders and they breathe through their skin. So anytime it gets too hot, they have to go hide out underground. And anytime it gets too cold, they have to go hide out underground where it's warmer. And their optimum operating temperature is about 16 degrees Celsius. So that's spring and fall is the best time to flip a log and find a redback salamander. And that's because they really can't tolerate temperatures outside of that range. So in Appalachia, where temperatures are cool and it's just the conditions are right for the majority of the year, these animals really do thrive.
All right. We're going to go backwards. There's some questions about the chytrid fungus and BD. It says, Steve asked, how come invasive cane dogs are not affected by fungus and continue to spread across Australia?
So the question was cane toads?
Oh, maybe it was cane. Is it cane toads? Steve? Or because it says cane dogs, maybe it's cane
No,
toads.
cane toads. It's spelling correction.
Cane
I'm
toads.
sorry. Cane toads.
Sorry. Cane toads are not affected by
Spell
the
check.
fungus and can
It's
spread.
not okay.
Yeah, so some species are not susceptible to the amphibian chytrid fungus. One of those is the red-eyed tree frog. One of them is cane toads. Another one of them is the common bullfrog. They are what we call the typhoid Marys. They can get infected with the disease, take the disease into a community and spread it, but not die from it. And the bulk of this, when I look at just the total number of amphibians in Panama that are at risk of extinction from the amphibian chytrid fungus, probably about 50 species. About 9 of those have already gone extinct. And then of the remainder, we have secured captive breeding populations of 12 of them. But that still leaves about 150 species that are either too rare to have any idea, or they seem to live with the chytrid fungus just fine. So there are some frogs that can handle it a lot better than others, and that's understanding that I think is going to be the key to figuring out how to find a solution.
And piggybacking on that, we have a question. My 8-year-old would like to know: do the pathogens, do the fungus affect axolotl populations because they are amphibious? They might have an axolotl as a pet.
So axolotls are a very interesting species. They are called Ambystoma mexicanum, and our spotted salamanders in the United States and in Maryland are called Ambystoma maculatum. So they're in the same genus as our local spotted salamanders, right? The difference between spotted salamanders and axolotls is axolotls are pedomorphic. So that means that they never change from a tadpole into a grown-up salamander, whereas the spotted salamanders do change from tadpoles when they get to about that big. And then they get their spots and they move on to land and they become land-based animals, whereas the axolotls will breed in the water. Now their home is Lake Xochimilco in Mexico, which is basically the lake that Mexico City has been built on top of. So there's very little habitat left for axolotls in the wild, yet they're one of the most common species in captivity, and they are a model that's been used for a lot of research into like limb regeneration and eye development. And so those animals are actually in danger in the wild, in danger of extinction, but you can buy them in the pet store and they're very common. The question was, are they susceptible to the amphibian chytrid fungus? I do not believe that they're super susceptible to the chytrid fungus. I do know that our spotted salamanders do not seem to become infected with the chytrid fungus.
All right, and we have, there's a question from Alan. How many frogs are needed to maintain genetic diversity? And I think that you kind of touched on that with the 20 and 20, but which I thought was so low. But
well, it is low, but we want to bring that up as quickly in one generation. We want to bring that up to about 300 individuals. That's like the minimum amount, right? Ideally, you want a lot more than that. But when you're doing triage, you kind of just got to take what you can do, right? So our goals for our project are: we want 300 adults represented from 20 male and female founders for each of our species. And that should preserve 90% of genetic integrity of the species over about 100 generations.
that's quite amazing. Erica asks, are there any resources that you recommend for identifying salamanders, seal and dusky salamanders, etc.?
Mm, there's a couple of awesome books. There of salamanders of the eastern United States. There is just a new one that I have ordered. um, uh, but I have not got it yet, so I can't vouch for it. But there's some awesome field guides.
I have a question. You were talking about the hellbenders and how they create nests with eggs. Has there been any experimenting? Can you go into their nests and extract the eggs and then do them and breed them in captivity, like has been done with the bringing back the American alligator, etc.?
Yeah, so they have done what we call head starting for hellbenders, where they do go and get wild eggs and raise those in captivity. And that has been the foundation of many of the captive breeding and release trial programs. I believe the St. Louis Zoo has managed to breed hellbenders in captivity from scratch. So the eggs were laid and fertilized inside an exhibit, but they're the only ones that have done that. All of the other hellbenders in captivity have been from wild caught eggs, and so and the hellbenders that we used for our vaccination trial were from a clutch of wild collected eggs from the Buffalo Zoo.
How do you keep your moving on in a positive direction with all the negativity around the decline of amphibians, and how do you keep from getting subsumed by despair?
Well, you have to be optimistic, and I think that humans are really good at solving disease problems. If you see how quickly we managed to come up with the vaccination for COVID, if we put the right resources into the equation, then we can generally achieve a lot, right? And we have put a lot of resources into this, and we've learned a lot about the amphibian chytrid fungus. So I am hopeful that we will be able to find some solutions and get these animals back into the wild. But it doesn't, gloom and doom aside, it is a huge weight of responsibility, right? To have these creatures, and if we've got some issues right now where we had a corporate sponsor was shut down by the government of Panama. So we are, we've got a huge budget hole right now, and we've got to figure out how we can fill that, because if we don't, these species are just they're just gonna be wiped out. And I think it's a lot easier to figure out solutions to those problems than to try and pull these species back from extinction. So it's something we can do. It's something that we have the power to do. It's just something that a lot of people are really keen. There's a lot of very keen young biologists who really want to get into amphibian conservation, and they want to make a difference. And we've got to create the space for them to do that and help us to solve these. And I think that our human ingenuity has shown time and time again we're great at solving diseases in livestock and in humans, but not so much in wildlife. And I think that is going to change, in part because some of our efforts.
Any other questions for Dr. Gratwicke tonight? If not, thank you so much. This has been wonderful. And I love ending on a positive note. I love the fact that we can get out there and dig a hole in our backyards and create amphibian factories right where we live. So I hope that we do that. And we go out there, we have field trips to see the vernal pools every spring with our organization. And it's a magical experience if you can catch it on the right night and the mating of the spotted salamanders. And thank you for all that you do and with your team and everything. It seems like that you were hopefully in good hands. And we just appreciate everything that you're doing to help sustain and conserve these wonderful pieces, these wonderful art pieces of our natural world.
Well, thank you so much for having me, and thank you for allowing me to talk.
All right, thank you so much. Everybody stay well, stay curious, and stay outside, and we'll see you next week for more Must Learn Thursday with the Natural History Society of Maryland, or come visit us on Sunday when we'll learn all about vertebrates at the museum. Take care, everybody. Bye-bye.
Night.